You guys are welcome to sit if you'd like, but I understand why you might want to stand back there. Welcome to the Boardwalk Talks series here at the Alabama Aquarium at the Dauphin Island Sea Lab. My name is Mendel Graber. I am one of the educators here at the lab. And today we are going to chat with Lydia Hayes Costella, who is going to talk to us about marine deep subsurface. So these are organisms that live below the seafloor in the deep ocean, fungi, and maybe some other deep subsurface cells and microbes. So with that, I am going to let her tell you about these microbes and about some of the incredible challenges associated with collecting them and exploring the deep sea subsurface.
Great. Thank you guys all for joining. I hope that you enjoy what I have to share with you. So I'm Lydia. I am a PhD student here at Dauphin Island Sea Lab and the University of South Alabama, and I study geomicrobiology in extreme environments. And as a geomicrobiologist, I study basically how microorganisms interact with geological and geochemical processes and how things like minerals and metals can affect microbial growth, their activity, and their ability to survive. So I study life in the marine deep subsurface. Like what Mendel said, it's the organisms that live far below the seafloor. So we're talking 10 to thousands of meters below the seafloor. So think the average depth of the ocean is between 3,500 and 4,000 meters. So then we're talking hundreds to thousands of meters below that. So this system is considered extreme for a number of reasons. One of them is nutrient and energy availability for life. It's extremely high pressure. There's a wide range of pHs, going from extremely acidic to extremely basic, as well as the possibility of metal toxicities and radioactivity, among a lot of other extreme conditions. So we study what microorganisms can exist in these environments, what they're doing, and how they're able to survive those conditions, even when you think about how hostile that might be. You might think life could not possibly exist there, but in the wise words of Jeff Goldblum in Jurassic Park, life finds a way. So why do we care about life in the deep subsurface? In 1 milliliter of seawater, there are 1 million prokaryotic cells, so that's bacteria and archaea, and 10 million viruses and thousands of eukaryotic microbial cells. So there are more microbes in 1 milliliter of seawater than there are stars in the sky. So I have a picture here that I'm going to pass around. It is a microscope stain of 1 milliliter of seawater, and you can see how many cells there really are. So in this 1 milliliter of seawater, there's millions of cells. And it's estimated that in the entire sub-seafloor, there are the same amount of cells as there are in the entire ocean's water column. So you can imagine that there's a lot of life down there, even though it seems like nothing could survive. And that life is really important for geochemical cycling, like carbon cycling, sulfur cycling, nitrogen cycling, all of these things that we as people need to live. And so it's really important to understand what they're doing and how their role impacts these cycles and how it affects our lives. So microorganisms have a lot of different survival strategies that enable them to survive in these environments, including using a lot of different types of metabolisms. So for instance, we have microbes that live in the soil, and this is called a mudwat. It uses energy produced by microbes oxidizing iron, and it produces energy. They're called geobacter. And it's kind of hard to see in this lighting, but there's actually a light that is lighting up on both of these every 30 seconds or so. That is produced from energy from the microorganisms. Another survival strategy is the production of antimicrobials, or in counter to that, antimicrobial resistance. And then additionally, the ability to go dormant or produce spores. That allows them to wait in these subsurface environments until the correct ideal conditions allow them to wake up and be active again. So when people think of microbial life, they often think of bacteria and archaea, the things that make us sick, the things that, I don't know, they do a lot of things. But they often forget about fungi. Fungi we think of usually as mushrooms, but there are also microbial fungi that you can't see with your naked eye until it's in a much larger amount. And fungi are important in the deep subsurface because of their role in carbon cycling. They have the unique ability to break down what's called recalcitrant carbon, which is really just a big term for carbons that are hard to break down. And they have the ability to break those down into smaller pieces so that other microorganisms can actually use those carbons. And for example, we get energy when we eat cake. But if we chew our fingernails, we're not going to get any energy. But fungi can get, even though both of those things are made of carbon, fungi would be able to get energy from eating fingernails. Keratin is a type of recalcitrant carbon, whereas cake is a labile carbon, or sugar is a labile carbon. So it's easier for other organisms to gain energy. So in the deep subsurface, where there's very limited resources for energy, fungi are able to break down the things that other organisms can't and get energy from that, and then give back to the microbial community more carbon in a form that they can eat. And they also are able to provide carbon sources when they die and they become necromass in the sediment. So these are really important in these nutrient and energy limited environments, but unfortunately we don't know very much about them because sampling the deep subsurface is really challenging, both logistically and it's very expensive. So how do we sample these environments? It oftentimes requires deep sea drilling. So there are a few different ships under the International Ocean Discovery Program and its previous iterations that have expeditions that go out on these very detailed, logistically complicated expeditions to collect samples from the deep subsurface, drilling as far as, I think the deepest hole that's ever been drilled was 3,000 meters. Below the seafloor. And like I said, it can be very, very expensive and challenging. But basically how these samples get drilled is I have a little demonstration. So we have our seafloor sediment. If I can take this container apart. And the drilling vessel will drill a hole with a drill string and a core liner. And the deeper you go, the more sediment you're able to core. And then we can also collect fluids from once these cores have been drilled by installing what are called corks. It stands for Circulation Obviation Retrofit Kit. And we can install these devices and send tubing down, which will allow us to pull up fluids from deep below the subsurface. So I have a couple of pictures to pass around that goes along with that. So this first picture is a picture of what the corks look like in the holes in the subsurface. And this second photo is a picture of tubes going into the cork.
That's a neat demo. That was a neat demo.
Thanks. Yes, so once they're installed, they are left in place. And they don't get installed into every hole that gets drilled, but certain holes, they are installed and they are left in place, which allows scientists to go back and access those same holes to do future experiments or continued observations.
That's amazing to me. They can hit the same. The ships can hit the same.
Same
holes,
spot.
Yeah, to repeat that.
How big are the holes on the seafloor? That's insane.
Like the cores that come out are like this big, but the cork hole is about this big.
And how big are the ships that try to find those again?
Really, really big.
And how big is the ocean?
And how big is the ocean? Even bigger.
Okay, one of those.
And the cores that come up, they look about like this, except for they come up in sections that are 9.5 meters long, so much taller than this. Additionally, we sample surface sediments, not using IODP's operations, but another way we can sample the deep subsurface is using other types of drilling, or not drilling equipment, but other types of coring equipment. This was taken with what's called a push core, which was the third image that I passed around. But yeah, so this is how scientists actually go and sample these environments.
Lydia, how can you be sure that the microbes that you're finding several meters down, in your core, that would indicate that they're several meters below the sea floor, have not been pushed there by the core itself, and so they're present in that depth from being pushed from the surface by the coring operation?
Yeah, that's a really great question. So we utilize a lot of different controls to account for that. So when the drill vessel is drilling a hole, it uses drilling fluid to keep the rotor cold enough to be able to drill through, because friction makes things warm, right? And so we collect that drilling fluid and we'll sequence that alongside our sampled organisms and make sure that the organisms that are in that drilling fluid are not present in our community. Additionally, we use bottom water samples, which is water samples that are filtered from the bottom of the seafloor, to make sure that the samples that the community we're looking at isn't from the seawater, it's actually from the sediment. And when we actually sample from our core, we take it from the middle of the middle. So when we actually collect the sample that we use to extract DNA and RNA to look at our organisms, we use syringes or chisels to actually access the middle part that hasn't touched the outside. So then we can ensure that what we're working with is actually from where we say it's from. Yeah, so once we collect these samples, what do we do with them? We approach the search for fungi in a multitude of ways. The first way is called culture dependent, which means that we actually take the sediment and we bring it to the lab and we put it in media, which is basically like soup for microbes, and we can grow them physically in the lab. And what that allows us to do is isolate a very specific organism, and we can test its ability to survive under extreme conditions. If we can test in the lab different pH levels, different temperatures, different salt content, different sources of carbon, those kinds of things, and just see what are the limits of life for that organism in these different conditions.
Are there any of those conditions that you would consider extreme that you need to replicate in order for them to thrive and survive and culture them?
Yes, sometimes. So that's one of the challenges about culturing in the lab, is that not everything will grow under every condition. So sometimes you'll find that you might not grow anything if your conditions aren't right. It's kind of, it's really rewarding to be able to culture things in the lab, but it can be really challenging as well. And so sometimes you have to, it's kind of like Goldilocks. You have to find what's just right. And then,
are there kind of protocols that are already established that help you succeed at culturing these, or is this kind of something you're learning as you go?
a little bit of both. So there are definitely established procedures for culturing in general, but every environment is different, so you have to take in different considerations for each thing. So there is kind of a basic, like, you know that everything needs carbon and everything needs nitrogen in order to survive, but how much of everything and what type is kind of a guess and check. And then additionally to culture dependent, we have culture independent methods for looking at life in deep subsurface, which includes looking at the DNA, which gives you the organisms that are there, but they might not be alive or active, and then looking at RNA, which will tell you what organisms are actually active and what they're doing. And that is all done through sequencing and genome sequencing and metagenomics and metatranscriptomics. And so that will tell you about the microbial community as a whole in each of these environments.
What do you have in your jar there?
Oh yeah, and this is one of my deep sea fungi growing in this jar that I've been able to culture.
Where'd it come from?
This one came from the South Atlantic transect, which is the deep subsurface beneath the South Atlantic gyre. It's an area.
Something we can pass?
Yeah. It is an area where there is very little carbon, really low nutrients, and this one came from 105 meters below the seafloor.
Is there a lot of diversity of these organisms, or are there, from place to place? So are you going to find a lot of the same species, say, in that location and also in specific sampling sites, or?
There is diversity and there is overlap, so it can go both ways. I have fungi that I'm sampling in South Atlantic transect and also in the Mariana Forearc, and some of them overlap and some of them are unique. So,
how many have been identified? Do you know? Like, how many different species?
In the subsurface, or a lot? I do not know how many though. Yeah.
Out of your fungi cultures, what's the deepest one that you have?
That one is the deepest one. That's 105 meters below the seafloor. That's not the deepest that has been cultured, just personally that I've cultured.
Are any of these organisms affected by pressure, or is it just completely different for them?
Yeah, definitely. They are affected by pressure. In fact, that's that can be one of the challenges in culturing them in the lab, is they might not make it up. They might not survive.
I'm so curious about that.
Since you referenced Jurassic Park, is there any biosecurity concern that you're going to wake up something that's
To be honest, most things in the deep subsurface can't survive in temperatures that would affect humans, so they're non-pathogenic. Yeah, nothing that survives in those cold, extreme temperatures are going to be able to be a host or hosted in the human body.
Ways in which the South Atlantic transect microbial community
Can you repeat the question?
Can you repeat the question?
Yeah. So I was just curious about how the microbial communities overlap with one another compared to the Marianas and the South Atlantic.
Tell us a little bit about the different cruises that you've done, and then you can answer her
Sure.
question about any overlap.
So the two projects that I'm working on, one of them, like I said before, is the South Atlantic transect, that area of sediment below the South Atlantic gyre. I was not actually on that cruise. The samples were shipped to us. But I was on the cruise to return to the Mariana Forearc, which is off the coast of Guam in the Pacific Ocean. And the question that we just got was, what are the overlaps in microbial communities between those two sites? And I can tell you honestly that I don't know, because I have not gotten those sequences back yet. Any other questions?
What do you find most interesting about what you're learning?
What do I find most interesting about what I'm learning? Honestly, the different survival strategies that microbes have in order to be able to withstand different extreme environments.
So could you describe some of those, or are those still a matter of trying to understand how they're doing that?
So the specifics to the organisms I'm working with are still vague, since I have not received the sequences yet in order to dive into that. But in general, they have a lot of different survival strategies, like their ability to, for instance, in the Mariana, the environment is really extremely high pH, above 10, whereas water is 7, or ocean water is about 8.1. And in order for microorganisms to be able to tolerate or thrive in that environment, they have to be able to keep their insides of their organism at a lower pH, but be able to keep the outside tolerant to the higher pH. So they have to have a proton pump that allows them to keep that stable balance. So that's one survival strategy. They also, organisms can also be dormant and form spores when conditions are extreme and they are not optimal for life.
What might be a condition that would stimulate that, and then what would prompt them to activate?
So if they do not have enough carbon to survive, then they can go dormant. Something that might stimulate them to revive, which might not ever happen in the deep subsurface, but they can wait and wait and wait on a geological timescale. But the influx of water from basement fluids into the deep sea sediment can provide nutrients or carbon from environments that don't have enough of it, and that could stimulate the, awakening of a dormant spore.
Something you just said that was really interesting is you said these things can lay dormant on a geological timescale.
Yes.
What's the longest period, or what's a period of dormancy look like on that scale?
Thousands of years.
Thousands of years. What's the longest that we know? Do we know?
I do not know.
Okay, that's incredible.
So you are talking about the sequencing, the genomic sequencing. So there is a catalog of these organisms that have been identified by sequencing, and so when you send samples to be sequenced, they can be compared against a known catalog.
Yeah.
Okay, and so then they can be matched to a known species or identified as an unknown species?
Correct. So yeah, there are all sorts of databases online that once we get those sequences analyzed, we can annotate them, which is basically identify what all of the genes are compared to known organisms. And we can also compare the community as a whole and all of the organisms that we find in that DNA to known organisms, and determine whether what we have is unique or whether it's something that has been found in other places.
So you were talking about their importance in the nutrient cycle and how this can happen on a shorter time scale. This nutrient cycling, for example, organisms that are eating and moving nutrients around, or we can talk about it on a much longer time scale where you're talking about compounds that are getting kind of buried in the seafloor, and then over geologic time, when we talk about geologic time, we're talking about could be millions of years or more. These through like plate tectonics, for example, these sediments are being moved around, and so our Earth, part of the reason our Earth is a living Earth is because we have that tectonic movement. So it is cycling these rocks, these minerals, these compounds around the Earth. Otherwise, they might all just kind of like get buried and sink into the core of the Earth and never resurface. So these organisms are playing an important role in that. Do you expect that there are things that humans are doing, like human activities? We are kind of familiar with the human activities that might be affecting our atmosphere or the ice. Are there things that humans are doing that you might expect to impact this process?
So there are a lot of things that can impact life in all sorts of different environments. One of the things that could potentially impact fungi or life in general in deep subsurface is changing in atmospheric CO2 concentration, because that leads to the acidification of the ocean and the amount of carbonates that are settled into the sediment column, which will affect the different carbon availability in the sediments.
Can you see any of that, or would that be too kind of fine a resolution for you to see on this kind of time scale?
On our time scale, it's probably too fine of a resolution, but you can see changes in CO2 or in carbon in sediments over geological time, and you can use that as a proxy.
So if you were to kind of give folks a takeaway message, what would you want them to take away understanding?
That organisms in general, microorganisms in the deep subsurface, are really interesting. They do a lot of things that we can't even imagine yet in order to survive, and they aren't very well studied, and we should continue studying them.
Thank you.
Thank you all for listening. Oh, one more question? Yeah, no worries.
I was just curious, about how long have people been taking samples from the subsurface? Like, how long have these projects been going?
So the International Ocean Discovery Program and its previous iterations have been around for about 60 years. But for microbial purposes, it's been about 15 years.
It's a pretty new area of research.
Yeah, it's definitely up and coming.
So there are a lot of challenges associated with creating these, baseline information against which to compare. So, it sounds like there's more than I thought there might be, as far as that research baseline to go on, considering it's a new science. But
yeah, so I'm going to ballpark it, there's about 12 or so sites within IODP's sampling regime that has sampled for with microbial science as a goal in their scientific objectives, but only 5 or 6 of those have focused on fungi. So it's still pretty new. There's not that much to go off of.
so how many sampling sites are there for the drilling?
That's a good question. Do you know? Thousands of holes.
And only a few of them are. Is anyone looking at the fungal community?
Yes.
Interesting.
So I've heard the International Ocean Discovery Program compared to NASA. First, do you think that's a proper comparison? And I think I would think so. And second, how often do you guys do these cruises, and how often are you able to get this information, especially since we don't know that much about it?
So NASA and IODP are different things, but they are kind of linked in ways that, like deep sea subsurface in studies can be linked to astrobiology studies. So they do kind of have that sort of overlap. And as time is going on, the scientists involved in both of those organizations do collaborate and work together under different circumstances. I forgot the rest of your question.
How often are you guys able to get these samples? That was me.
The main question is how often. Up until this year, there's usually a couple of expeditions a year. They are 2-month-long expeditions, so it takes a lot to mobilize and demobilize those ships, where the IODP ships, they have basically most of the science equipment that you need for them to do biogeochemical studies, paleo studies, right on board to give you on-ship information as you're sampling to influence your sampling objectives. So it's basically a lot of science labs in shipping containers that get stacked onto these ships and go out to sea. And it can take a lot of effort in order to do that. So they only have a few a year that can go out. Unfortunately, the JOIDES resolution is being retired this year. So we're not entirely sure what the next steps are going to be.
So how long does it take to process? I mean, I guess you find a lot to do with the samples that you do get to take. Excuse me. And these, limited opportunities to go out and take samples. So how long does it take you to process the samples that you're getting?
Well, I have had my samples for 2 years, and I have not gotten my sequences back yet. It depends on several factors. One of them that I'm struggling with is that the biomass, so the number of cells in my samples are so low that I have to extract and extract and extract before I even have enough DNA to get a sequence. So that's one of the challenges that I face. So it really depends on the environment, how difficult that is going to be. And so far I'm on year 2 and we're getting there.
So is that due to difficulty culturing them?
So I was pretty successful in culturing. From South Atlantic transect, I was able to grow 25 organisms from different samples, which is quite a lot. But culturing and metagenomic and metatranscriptomics are kind of 2 different forks in the road. And so culturing is, if you can culture things, it's a lot easier to get that DNA because you have it growing actively. Whereas when you're looking at DNA in sediment and not culturing and you're just trying to extract that DNA straight from the sediment, you have a limited number of cells to work with. So you get less.
So you were explaining how rich they, have the high density of the microorganisms. You used a milliliter of water as a standard. So why would you have such a low density in your sediment sample?
They are not evenly distributed. So in areas that are really low carbon, low nutrients, there's going to be less activity, so they're not going to reproduce as quickly as they might in the shallow sediments or in coastal zones where they're getting a river runoff and all sorts of nutrients that they can survive off of.
So So they're not necessarily, there are hot spots for them, even though there are a lot of them in the marine subsurface. There are kind of hot spots like there are for other kinds of organisms.
Yep.
And so you aren't necessarily hitting those in your sampling spots.
And it's not necessarily that we aren't hitting them, because they are there. It's just that the number of them can be really small.
Where's your sample from that you brought?
This sample? This was taken just off the dock.
Oh, okay. Well, thank you for coming in and sharing. That's a pretty interesting area of emerging research, and it has some really interesting challenges and applications, and a lot of room to grow.
Definitely, it's definitely a work in progress for everyone. But yeah, thank you for having me, and thank you all for hanging around and listening to my talk.
You guys can come closer, I think, if you want to come see the. I've been seeing.